Imagine standing in your kitchen, looking around, and thinking you can see what is there. The counter, the toaster, a banana entering the final, deeply personal stage of decay. But you are not seeing the whole kitchen. You are seeing a heavily edited version made from the tiny range of information your eyes can detect and your brain considers useful. There is light passing through the room that is invisible to you. Microorganisms occupy surfaces that look perfectly empty. The toaster is radiating heat in wavelengths your eyes cannot register. Processes are unfolding too quickly or too slowly for your brain to notice. The strange truth is that human brains were never equipped to see most of reality. Not because reality is hiding. We simply arrived with a very limited viewing package. Vision feels direct, like the eyes are windows and the brain is a small person watching through them. In reality, your eyes collect a narrow stream of signals. Your brain then constructs a useful model from that stream. Useful is the important word. Evolution did not need to produce an animal capable of viewing every wavelength, bacterium, magnetic field, and geological process. It needed an animal that could find food, recognize faces, judge distances, avoid predators, and notice when another human was holding a suspiciously large rock. Human brains became remarkably good at processing information, solving problems, and creating abstract ideas. But large brains are energetically expensive. Biological systems involve trade-offs, and perfect perception would be an extravagant luxury. So natural selection gave us senses fitted to human-sized problems. Start with light. What we call visible light is only one section of the electromagnetic spectrum. NASA generally places human vision between wavelengths of roughly 380 and 700 nanometers. Within that narrow band, different mixtures of wavelengths become the colors your brain labels violet, blue, green, yellow, and red. Outside it, the spectrum keeps going. Past red lies infrared, followed by microwaves and radio waves. Past violet lies ultraviolet, then X-rays and gamma rays. These are not separate magical substances. They are forms of electromagnetic radiation with different wavelengths and energies. Human eyes simply do not contain receptors capable of handling most of them under ordinary conditions. This creates a slightly embarrassing situation. You can stare directly at a powerful radio transmission and see absolutely nothing. Your Wi-Fi router can fill the room with signals while sitting there looking like a disappointed plastic crab. Infrared is especially interesting because warm objects emit it. Your body, your coffee, and the motor inside your refrigerator all produce infrared radiation. A thermal camera detects part of that radiation and maps different measurements onto visible colors. The glowing orange person in a thermal image is not literally orange. The camera is translating information your eyes cannot receive into a visual language your brain already understands. Ultraviolet reveals a different world. Many flowers reflect ultraviolet patterns that can guide pollinating insects. To human eyes, the flower may look evenly yellow. To an animal sensitive to ultraviolet, it may contain high contrast markings pointing toward the center. Neither view is the one true appearance of the flower. Each nervous system receives a different sample of the same physical object. And if you enjoy having familiar reality quietly removed from beneath you, subscribe to Oddly Human. We spend a surprising amount of time discovering that normal life is not especially normal. Our limited spectrum also explains why telescope images require careful translation. The James Webb Space Telescope observes primarily in infrared. Its famous images are not simple snapshots in the way a phone photographs a birthday cake. Webb records data through different filters. Image specialists then assign visible colors to those infrared wavelengths, usually preserving their order by mapping shorter wavelengths toward bluer colors and longer wavelengths toward redder ones. This is sometimes called false color, which sounds suspicious, as though astronomers have been caught giving space an unauthorized makeover. But false color does not mean fake data. It means translated data. The color choices allow human eyes to distinguish structures, temperatures, materials, and wavelengths that would otherwise remain invisible. The result is not exactly what you would see while floating beside a nebula. It is closer to a scientific diagram made from real measurements. We do the same thing whenever information falls outside our senses. Weather maps convert temperature into color. Medical scanners turn radio signals or X-ray measurements into images. Geiger counters turn radiation into clicks. NASA has even converted telescope data into sound, mapping features such as brightness and position onto volume and pitch. Science is full of machines that say, in effect, you cannot sense this, so here is a version you can. Wavelength is only one limit. Size is another. Look at your hand. The skin appears continuous. Under magnification, that smooth surface becomes a landscape of ridges, pores, flakes, fibers, and microorganisms. Go deeper, and tissues become cells. Cells become complex structures made from membranes, proteins, and molecular machinery. None of this is naturally visible because our eyes have limited resolution. If two tiny details are too close together, their light blurs into one feature. Magnification can make something look larger, but useful microscopy also needs enough resolving power to separate the details. Light microscopes extend our vision by using lenses and carefully controlled illumination. Electron microscopes go further by using beams of electrons rather than visible light. Detectors measure how those electrons interact with a specimen, and the measurements are assembled into an image. Again, the final picture is a translation. Those spectacular microscopic images of viruses, pollen, and insect faces are often colored afterward because electron microscope data do not arrive as ordinary visible color. The color may separate structures or simply make the image easier to interpret. This does not make the tiny structures imaginary. It means color is being used as a label. Human perception also occupies a narrow window in time. Some events happen too quickly for us to separate. A rapidly spinning fan becomes a transparent disk. Individual flashes can blend into steady light. A hummingbird's wing turns into a blur because the visual system must gather information over short intervals rather than produce infinitely fast updates. Slow-motion cameras borrow a faster sense of time. They record many images in rapid succession, then play them back slowly enough for the human brain to inspect. Suddenly, a balloon does not merely pop. Its skin tears, curls backward, and abandons the water for a brief moment before gravity notices. At the other end, some processes are too slow. Plants turn toward light. Fungi spread. Shadows travel across buildings. Coastlines erode. Stars move through the sky. Your face changes across decades while appearing basically identical every morning, which is either comforting or an impressive administrative failure. Time-lapse photography compresses these slow changes into our perceptual window. It does not create motion. It reveals motion that was always happening at a pace we could not experience directly. This is a useful correction to the phrase, "seeing is believing." Seeing depends heavily on scale, speed, wavelength, contrast, and translation. Something can be real and still be invisible because it is too small, too distant, too faint, too fast, too slow, or expressed through a signal your body cannot detect. There is also a deeper limitation. Even inside the range your eyes can receive, the brain does not preserve everything. You have a blind spot in each eye where the optic nerve leaves the retina. You normally do not notice a hole in your vision because the brain fills the region using surrounding information and input from the other eye. Your brain also stabilizes the world while your eyes jump rapidly from point to point. It adjusts for changes in lighting. It emphasizes edges and contrast. It directs attention towards some objects while allowing others to disappear in plain sight. Vision is not a complete recording. It is an active prediction about what is probably out there. That is usually a strength. A brain that inspected every photon with equal interest would be terrible at crossing a street. We need compression. We need shortcuts. The problem begins when the edited version feels complete. Recent research makes that boundary especially clear. In 2025, researchers reported experimental contact lenses containing materials that converted near-infrared light into visible wavelengths. Human participants could use the converted signals to distinguish infrared information that would normally be inaccessible to them. The lenses did not teach the retina a completely new form of vision. They performed translation before the light reached it. That is the larger human trick. We were not born able to see bacteria, infrared heat, distant radio galaxies, X-ray structures, or a plant moving through an afternoon. So we built microscopes, sensors, telescopes, cameras, scanners, graphs, and algorithms. Then we converted the results into colors, sounds, shapes, and movements that our ancient visual system could understand. Our senses may be narrow, but our ability to extend them is extraordinary. Science is not just a collection of facts gathered by unusually patient people. It is a system for borrowing senses we were never born with. So the next time you look around your kitchen, remember that the room is not empty, still, silent, or fully visible. That is simply the version your brain can open. If there is another hidden part of everyday reality you want us to explore, leave it in the comments. It may be sitting directly in front of all of us, being extremely real and completely ignored.